So good afternoon, everyone. I am really pleased to have the opportunity to frame and start this conversation that we're having with the two co-chairs of the Consensus Study Committee. So in September of 2021, NHGRI, with 13 other institute centers and offices at NIH, asked the National Academies to conduct a study focused on the use of population descriptors in genetics and genomics research. So we finalized that contract with the National Academies. In February of, it was actually Valentine's Day, February 14th of 2022, the committee had been convened by the National Academies and had its first meeting with representatives from NIH to hear more about the charge that was provided them and for NIH to answer their questions. In March of this year, the committee released its report focused on the use of race, ethnicity, and other population descriptors in genetics and genomics research. So we're so pleased to have the two co-chairs here to present information about the report, and hopefully we have an engaging conversation with council about the report. And so I am pleased here to introduce people that we know well, Dr. Aravinda Chakravarti, who is the director of the Center for Human Genetics and Genomics, and the Muriel G. George Singer, professor of neuroscience, physiology, and professor of medicine at NY School of Medicine, and Charmaine, Dr. Charmaine Royal, who is the Robert Cohane Professor of African and African American Studies, Biology, Global Health, and Family Medicine and Community Health at Duke University. And she also directs the Center on Genomics, Race, Identity, Difference, and the Center for Truth, Racial Healing and Transformation at Duke University to present the report and to have a conversation with council.
Okay, good afternoon. So I should begin with some thoughts and some thanks. And I think what Charmaine and I will try to do is sort of lead you through the thinking in this report. What we and 15 others did took about a year or more, and I'm not quite so sure you want to hear every single minute of that conversation, but hopefully what both of us would be able to do is distill it down into the thinking that not only we developed, but rather what hopefully each of you will think through as well. So first, some thanks really to NHGRI and all of us, and I believe there were 13 or 14 other institutes that championed this cause. And there's no doubt that Eric Green, as well as Vince Bonham, played very important roles in bringing this question to the front. There are many people in the National Academies that also should be thanked, and you should look at the report for all of them, but in particular, Dr. Sarah Beachy, who was really the chief protagonist on that side, and leading the report to the report, if you wanted to, you could see in your hands. It takes a lot of cooperation and collaboration to get it there, a lot of hard work, and Sarah put in more than her usual share. The last bit of thanks is actually to Charmaine. I should say Dr. Charmaine Royal, whom I've known for now a number of decades, and we've exchanged many views on this topic, but not quite in such a focused way. And so really both of us appreciate this opportunity to be able to do this and do this with other colleagues. Some, as you will notice from the membership, that some are geneticists all of you know and love, and others are social scientists, that is people whose work we've read, and yet others who were sort of new to the task. Okay, so I'm not going to read the statement of task, but I'm going to summarize it in the following way. I think the problem that we have is if we are to proceed with doing any kind of science, but clearly genetics and genomics, we need to describe the individuals who are part of a study and also need to rationalize whom we've picked for the study and why. Classically, this has been done often really using current norms and not without much thought, and race and ethnicity have really become common parlance. But we know through the studies of genetic variation in humans, particularly over the last 20 years, that there are much better, more scientific way to describe human subjects. And something not covered by our report is how we are going to assess really diversity and how we're going to compare diversity in individuals whose phenotypes we are studying. So the statement of task was to assess what kind of population descriptors we currently use and what we should use, particularly as it relates to studies of genetics and genomics. We need to develop approaches that can rationalize and advance the appropriate use of population descriptors and the kinds of best practices that we need to champion. We also need to discuss the obstacles to implementation, adoption of these best practices. Good ideas take a lot of work in order to bring them into practice. So here's a committee membership. I'm, again, not going to dwell on it because I already mentioned the fact that it represents a very broad sort of swath of science, and including other sort of measures of individuals that were important, that is, people who've been in some kind of research scholarship for decades, some who are much younger, and I think that kind of mix was very important for us to have new thoughts on this problem. So I think essentially one of the first things we had to do is to sit and think of what a population descriptor is. And there's no doubt this is a concept of a difference. And one of the other ideas that we analogize was like a concept of wealth. It can be measured in many different ways, but a population descriptor, there is not only one way. And yes, it is a way in which individuals can be classified and categorized into groups or populations, and populations here is in quotes because that itself could be defined in many ways. And I think that was a major sort of point for all of the committee to understand. And one of the second things that came out of those discussions is that the salience of any given population descriptor that we use is not fixed in time. This is not like other scientific definitions. And that this has varied from place to place and over time, and clearly it's varied across the globe. Now, to geneticists and those using genomics in the genetic context, a particular interest is, of course, descent-associated population descriptors. And these, of course, as you can understand, are populations whose members are thought to share some heritable characteristic from a common origin. And this, of course, can be described, it's not only one kind, it's a family of descent-associated categorization systems, and they also vary across the globe, and clan and caste and tribe and ancestry and ethnicity and others are part of these descriptions. So what's the problem with the current way in which we, and I should say, including myself, thought of the current use? The first is, we often lapse into typological thinking. We reinforce the misconception that human beings can be objectively grouped into, doesn't matter, 3 or 33 different groups, and that these groups are innate, that they're discrete. And this, of course, has been sort of violated by almost any study of genetic variation that we do, because despite our assumption, we constantly come across individuals, by any definition, are in quote admixed. That is, they don't follow any one single group label. The second is our continuing use of race. It is, of course, a scientifically invalid measure of human genetic difference because human genetic differences have occurred, a history that we now at least partially have uncovered through studies in genetics, and that race remains one of the most potent socially constructed groupings. Now, they may be useful in society, and in fact, they are useful in society, just as the OMB categories are, in order to assure that society makes progress on features that we've used to define people and denied progress to. But doing so in the scientific context needs to be completely rethought because it's harmful and erroneous and really not the best way to do genetics research. But there can be exceptions. Again, there's been a failure to realize that population differences also reflect differences in environmental exposures. During this committee, I often mention a very old population geneticist who was my mentor and as well as colleague, C.C. Lee, who as soon as I entered into facultyhood told me, he said, Aravinda, we are geneticists, not hereditarians. He was a plant breeder, and he knew that environments have very strong control over the genes that we have and inherit. So we always have to make sure that we choose individuals, and when we choose or ascertain individuals, be mindful of whether they are not only that the population differences are genetic, but that they could also involve environmental factors. And finally, there's, of course, been inconsistent definitions of almost any kind of descriptor that we've used. So there have been many reports that have pointed out the inadequate sort of use of the term race and ethnicity, or even strong use of that term in genetic studies. So why do we need another report now? I think there's no doubt that over the past 5 years, particularly, there's been an urgent need to address issues surrounding race and racism in science and society. And there's still a debate, at least in some quarters, as to how important this is, but there's no doubt that this is a problem we need to think through and solve as much as we can in the years ahead. Genomics data has clearly become more accessible and widespread. Again, I would say over the last decade, it's much easier to obtain, it's much less expensive to obtain on hundreds of thousands of individuals. So rethinking this problem is much more important today and in fact urgent. The third is that current large-scale genomic studies aim to include far more diverse individuals. And I'm not going to define diversity here. It could be done in very many ways. Simply because the more genetic variation we have, the more environmental variation we have, the much better we are at finding the causes of human phenotypes. And then there are many others that I could bring to bear. But most importantly, I would say that there are now advanced methods of analyzing entire genomes and genetic variation between humans that need to be taken into account as we move forward. Now, previous efforts haven't done so well because I believe, as you will hear from Charmaine, we haven't really thought of the full way in which we could implement our findings. Very briefly, the report has been divided into two major sections. The first section is far more historical, and in fact, it speaks about the various descriptors that we have. We purposefully did not engage in a very deep dive into the history of using race and ethnicity in human genetics, in fact, in biological or rather scientific research, because there's a well-heeled literature, and we cite many of those sources. What's more important, or became more important to the committee, are the last 4 chapters, which are really the recommendations. Our question was, if we are to change, what will guide our change? And if we are to need certain guides, which we established as principles, the guiding principles, then what are we going to search for to know that we are making change in the long run? What are the requisites? Chapter 5 then focuses on the guidance for the selection and the use of population descriptors in a number of genetic studies. We classified them into 5 classes. It's not an exhaustive class, but we thought that it represented the vast majority of genetics and genomic studies. And importantly, we devoted a chapter to implementation and accountability. So what makes this report unique is our thinking of the basic principles, the guiding principles under which we will change, the requisites that will keep us on track to making these changes, the kinds of guidance for researchers, and the kinds of specific implementation strategies and methods we would use. I would turn this over to Charmaine, who will lead you through the rest of the slides. But quite importantly, the change is for each of us to make individually. And I hope you would read the report and think of that as well. Charmaine.
Thank you, Aravinda. And thank you, Eric and Vince, for inviting us to talk about this report to the council and to everyone else who's listening. I want to start by just saying, Aravinda, I don't remember if you did talk about what was out of scope. Did you get to that? I'm going to start by talking about the things. There were three things that the Institute, 3 or 4, that were part of our charge indicating that they're out of scope for us, right? And one of them was looking at the use of race in clinical practice. Another was racism in science and genetics and genomics more broadly. And we were specifically asked not to give specific policy recommendations to the NIH, right? So there were these things that, as you listen to me talk about these things, you realize that some of these things are overlapping and are part of the conversation about how we use race, ethnicity, and ancestry and other descriptors in genomics research. We touch on clinical applications, but we don't delve into it. And so that's another area that requires, it's certainly related to a lot of what we came up with in terms of our recommendations, but just so everyone knows, those things are out of scope. So with regard to, I'm hoping that I'm going back. All right, let me go back to what makes the report unique. Aravinda talked about the diversity of our committee, and many members on that committee had been part of these conversations that have been going on for a long time about how we use race or not, and ethnicity and ancestry in population genetics or in genetics research as population descriptors. And so as we were trying to think about what are we going to come up with in terms of moving forward in this work, as we thought about our framework, we thought about some of the things that were missing, that we thought were missing, or that did not, were not emphasized as much in other recommendations that have been put forward. And in thinking about a framework, we thought about not a framework that didn't just tell us what to do. It wasn't just about how we do things differently. It's a framework that incorporated a new way of thinking. And the thinking about these population descriptors for us has a major role to play in how we use them. And so our framework, as Aravinda said, started out with these principles. And I'm going to talk about each of these categories, the principles, the requisites. Principles undergird all of the recommendations. Our recommendations are chapters 4, 5, and 6, which are the requisites, the guidance, and the implementation. And for the requisites are these things that we think are critical to changing the way we do our work, and these are things that also influence how we think about these descriptors. So for guiding principles, these are foundational and core terms that present a vocabulary. For us, it was creating a common vocabulary that researchers can use to do this work. And these are principles, they're ethical principles, and they're scientific principles, and principles that hopefully will get us to do more trustworthy science, which for us, trustworthy science is science that is both scientifically and ethically sound. So respect, beneficence, equity, and justice, respecting the people, the communities who have a stake in how they're described, the individuals and the communities. And we have these principles because we realize that the field is dynamic, and so we're not able to come up with specific approaches for every situation, but we need to think about things that guide us in making decisions about what to do in any given situation. And these principles undergird all of the recommendations. So when you read the report, you'll see these principles. We specifically call these principles out in our recommendations and explain how they apply in those recommendations. So respect, beneficence, thinking about the benefits and the harms, the potential benefits, potential harms of using population descriptors in genetics and genomics research. And our goal, of course, is to maximize the benefits, minimize the harms. And related to that is equity and justice. How will our selection of population descriptors facilitate inequities, exacerbate health inequities or health disparities, or perpetuate the existing ones, or create new ones, and being very conscious of how we think about that. Validity and reproducibility, we're calling on researchers to carefully evaluate their research objectives and assess the appropriateness of using population descriptors in their studies, and which population descriptors. We maintain that population descriptors are not always necessary, and we need to figure out when they are and which ones we might use. And transparency and replicability, having a clear rationale for using and selecting and using population descriptors and describing the process for making those decisions. So these are the principles that for us are critical to thinking about which population descriptors to use and how to use them. So we have these requisites that we thought of, and I'll talk a bit about them in a minute. And then for the requisites, we have typological thinking, measuring environmental factors, and engaging communities. And if our recommendations 1 to 5, these requisites are for a broad audience. And our thinking is that these requisites will facilitate the long-term success of these recommendations that we've made. And then chapter 5 is where we have specific guidance for researchers in terms of these studies, these types of studies, genetic studies that we talk about. And recommendations 6 to 8 are the guidances that we give to researchers. And along with those recommendations, we have best practices that give more specific guidance. And these are for researchers who are using genetics and genomics research, not only people who are formally trained in human genetics, but lots of researchers, as we know, are using genetics and genomics data. And then our final chapter, chapter 6, is about how we implement these recommendations. And we talk about implementation as being equally important to accountability, because accountability is what is going to at least increase the possibility that our recommendations will actually have some teeth, and that something will come of it, and that our report won't sit on a shelf. And so recommendations 9 through 13 are for the other members of what we call the human genomics ecosystem. And there are members of the ecosystem that support the work of the researchers. So typological thinking, and typological thinking is not always a bad thing. There are some cases where we do need to group and type different entities. But when we apply that thinking to human genetic variation, it gives us the misconception that human groups are biologically distinct, and these innate differences, and that these categories that can be grouped in these categories. We know that the patterns of variation, or human variation, are complex. And one of the other reasons for avoiding typological thinking is that it often includes thinking of homogeneity and discreteness, as well as hierarchical thinking, and that's where race comes in. Race is not just saying people are different and human populations are different. The hierarchy is a major part of the problem, and so recommendations 1 to 3 are about that. Recommendation 4 is about environmental factors. Aravinda talked about the fact that understanding the genome and our genetic variation is really incomplete without understanding the context within that genome operates. We make the point that descent-associated population descriptors are not good proxies for environmental effects. In some cases, we may use them because we may not have the specific environmental factors, but it's best to measure the variables we are trying to find. And then our third requisite is engaging communities and populations, and realizing that the misperceptions and miscommunication and inaccurate information about human genetic variation can be harmful to populations and the individuals and the communities with which we work, and can get in the way of progress with regard to research. So we're calling on researchers to engage experts from communities that they're working with to help think about how to describe these populations and help come to agreement about what is appropriate and what might not be. For guidance for researchers, Chapter 5 is what some call the heart of the report. I mean, some researchers will probably just gravitate to Chapter 5. Let me see what they're telling me to do, right? We encourage you to read Chapters 3 and 4 as well, because they will help you think about how to do what is in Chapter 5. But our guidance for researchers, our first recommendation is that researchers should tailor, this is recommendation 6, in the first recommendation in Chapter 5, is that researchers tailor their use of population descriptors to their research questions. This recommendation really is an overarching recommendation, really, because everything else in Chapter 5 is helping researchers to do that, is to tailor their use of the descriptors to the questions that they're asking. There are different types of genetic studies. On the right, you see we identify 7, it's really 5, but we look at Mendelian and complex traits in terms of gene discovery and trait prediction. So we have 7 types of genomic studies. There are others, but we saw these as key types of studies that we needed to talk about. So our recommendations 6 to 8 and our guidance for researchers are focused on these types of genomic studies. And health disparities, we're talking about health disparities studies that include genomic data, not all health disparities studies. So those that include genetic and genomic data. So we provide this table, and we created this table, the table. This table in Chapter 5, which is really a snapshot of a whole lot of text that's in Chapter 5. And we encourage anybody who looks at this table to read this table along with the text, because reading it by itself may not give you all the information you need to understand this text, to understand what's in the table. So what we did with this table is that we selected certain population descriptors. At the top, and in Chapter 2, we described these in detail. So we focused on these population descriptors: race, ethnicity, indigeneity, geography, genetic ancestry, and we also talk about genetic similarity along with those population descriptors. And we look at those in light of these types of genetic studies. And one of the takeaways is that race, as you see here, this is our legend. As you see, our advice is that these should not be used. Here with E is that if you do use them, they may be used as proxies for environmental, but not genetic. Question, in some cases, it may be okay to use these variables, and the plus is our preferred descriptors. And as you see, with race, for the most part, we discourage the use of race in any type of genetic studies, except for certain types of health disparity studies that are trying to understand the impacts of racism. Alternately, we suggest genetic similarity as a more appropriate measure of variation. Because we know that often and you see that for genetic ancestry, we have question marks here about what we do with genetic ancestry. We know that genetic ancestry is not directly observable. It is inferred. It is inferred from various measures of genetic similarity. So we're calling on researchers to be more specific about what they're looking at. Is it genetic ancestry that you're looking at, or is it genetic similarity? And in most cases, it is genetic similarity that we're looking at. And so genetic similarity, the quantitative measure of the genetic relatedness among individuals that relates to the extent of shared genetic ancestry. For definitions, so race, we say, should not be used except in the context of certain genetic studies, certain health disparity studies. And again, race we define as a socio-political construct, but we don't stop there. It's a construct that allows the classification and the ranking of human populations. But the reason that race is so problematic in genetics is that race, by its very construction, is based on ideas about biological difference. And so that is why it becomes important in our work and for us to clarify that race as used in genetic studies does not imply these innate biological differences. And that is why it's so complicated, because race by nature is based on those ideas. So for us, not using it is the best way to be clear and accurate and precise about what we're trying to do. And finally, our chapter 6, where we talk about how we implement these recommendations. And we identified certain members of the ecosystem, study participants, and helping study participants to become more vocal in terms of what they desire and working with researchers. Funders of genetics and genomics research, calling on funding agencies, as well as journals and professional societies to provide tools and educational modules for researchers for submission of grants, submission of manuscripts, review and submission of manuscripts and grants. Research institutions thinking about research institutions needing to build in incentives to facilitate interdisciplinarity. We know there are lots of obstacles to interdisciplinary work. And journals and professional societies, and the journalists and the media journalists as well as the media are the ones who communicate our science. And for us to be able to communicate with them more accurately what we're trying to do with, and hopefully that the communication that comes from the media and from journalists will more accurately represent what the studies are trying to do. So we're wanting scientists to get these descriptors and concepts right. And when we say right, we're talking about based on our principles, based on the requisites, being clear about what you're trying to do, ensuring that, that your use or not of population descriptors is directly relevant to the research questions that we're trying to ask. Geneticists and genomicists are collaborating and needing to collaborate with more, with researchers outside of those fields to help us have a more holistic view of health and of the conditions that we're trying to study. And we realize that it's going to take all of us, not just the researchers, but the whole ecosystem to change its way of thinking to facilitate the change that we all want to see. And I'll just close with that slide, resources from the National Academies related to our report. Along with the table is a decision tree that we created, and there's an interactive version of that also on the National Academies website for researchers asking specific questions and leading to specific answers and or suggestions about what they might want to do with their work. Thank you.
Vince, you want to come up? I'm going to ask Vince to moderate this discussion.
Oh, we stand here. All right.
Questions from council to start? Yes.
So thank you very much. I think this is really important as we're going forward. I'm going to ask the question of how this intersects with some of the other things that are going on, like the White House change in kind of the descriptors of populations, if this has any relationship with that, or also, the forms that we as researchers have to fill out at the end of our grants about different categories.
Great question. Thank you for that. I mean, what's happening at a national level in terms of this is we're still waiting to see some of what those outcomes are going to be, and it will invariably have some impact on what we're trying to do here. With regard to genetics and genomics, for us, what happens at the societal level is important for a lot of what goes on in society. When we think about research, I think we have to think about these labels and these use of these descriptors in different ways as they relate to our research and not. I think very often we conflate what happens in society, and they're related. But just like we say, there's some cases where the use of race and ethnicity and ancestry are useful for understanding certain outcomes and certain processes. And then there are times in research when they're not. And so I think regardless of what happens with regard to what is happening at in our country in terms of these policies, we still need to be clear about how those apply to us or not. And I think it will remain to be seen what we do in light of those. With regard to OMB, I'll let Aravinda speak, because the OMB categories, well, go ahead.
Yeah, I think the way that the committee thought about this is that a society may ask each of us who receives any benefit, say in this case from a grant, to do things that look apparently different or conflicting, but they aren't. So OMB categories are there to make sure that we represent our citizens sort of as best as we can equally in research. And I think in many ways, if you go back and look at the discussion on OMB categories, it's in that sense a political tool, political in the right way, meaning a good use of the term. But there's no need to assume that those same categorizations are helpful in a genetic study. So in a genetic study, we need to be able to look at the specific questions. That's why they're defined into 5 or 7 categories. And in many cases, I think really the bottom line of the committee was genetic similarity based on, say, reference samples, reference individuals. They're referenced, they're not idealized in any other way. That comparisons of any genome to them may in fact be sufficient, because that's basically what we technically do. As the number of genomes publicly available increases, and the best analogy I can find is each of us will look at a genome, just the way GPS works, with respect to 1 million other genomes. And I think ancestry is in fact a term that genetically is important, because we do inherit not only genes, but also culture. And so that brings up the whole matter that defining genetic variation far more in a granular way, that doesn't mean that it's done, the work is done. I think there's an important discussion as to how we construct reference, samples and individuals. But I think those kinds of ways would suffice what we do now and need to do in the at least immediate future. But I think there is an important discussion that we need to have, what comprises reference populations or samples, why, in what way do we sequence them, for what purpose? And there are a number of projects supported by NHGRI and others that are trying to tackle these issues, for example, in the Pan-Genome Project and others. So.
I had a couple of comments and a question. So the issue in epidemiology or genetic epidemiology is the lack of environmental measures. And I think we might correct that going forward with SDOH. But for legacy collections, that's difficult. And that's where race is helpful, because it is a measure of environment in some ways. The second comment was, how do we disseminate these concepts eventually to beyond the scientific community, to the in the end, this is an incredibly important task you're doing. You're defining humans. I think it has profound implications and how humans see themselves. And maybe this is so important that it has to go beyond the scientific community, and eventually has to be, the public has to be educated about some of these concepts. And my third question was what exactly do you mean by genetic similarity? What measure do you propose for that? Thank you.
So great question about environmental factors and legacy data. We touched on legacy collections but didn't go into detail because that requires a lot more work than was part of our charge. But we do, even in our decision tree, we do have questions for researchers about whether they're collecting new samples, or if they're using legacy data, and having specific questions about the variables related to those data and whether they need to incorporate those variables or not. So the questions that the researchers are trying to ask also will help them decide what they do with even the population descriptors that are part of legacy collections. How do they incorporate them? And also in terms of transparency, calling on researchers to talk about how they determined whether those variables are useful to them or not, and how they apply those in their own studies. The other question was,
Disseminate this beyond the scientific community. Did you think about that task? Although your immediate task was us and the genetic and the science community, but
our immediate task was the scientific community, and the issue of communication and science communication is something that we talked about as a committee, but didn't delve into that in our report. The implementation part of this, where we talk about the media and journalists, I think thinking about the media broadly and strategies to help the media and journalists in the popular media to understand what the report is really saying and what it's not, and to be able to appropriately communicate that to other entities. That will have to be part of the implementation, but was not part of our charge. But we recognize that we're not just speaking to the scientific community, we're speaking to the broader society. Even when we look at the requisites, our requisites are really for the broader society, especially the one about typological thinking and broader communities about how we think about human variation, right? We as geneticists, we talk about the fact that they're not these discrete groups, but how the public understands that. And we, from studies that we've done, see that the public doesn't have a great understanding of this. So all of that, I think, will have to be part of the implementation part.
Let me very quickly sort of add, I think we should think of a way forward. Legacy data, yes, are a very important source of the inferences we make today, but I think we need individual level environmental variables. I just don't think it's a well-designed study when we have individual level genomes, for example, on a whole group, however labeled, and then we use their average income, SES, and other average measures for the whole group as measures of environmental influences. So we need to think hard. I don't think the transition is going to happen in a day, but I think we need to think hard and go in that direction. The other is we have to, I don't know how broad to make this, but we have to describe our studies far better than we currently do to, for example, both individuals and communities whom we bring into our research. So we do need effective ways of describing. Yes, it's a much harder task than what we've done, but that's what we are up against. Up against meaning it is a challenge that we have to surmount. And finally, I think technically genetic similarity is basically all the technical ways that we do when you do principal components, it's actually measuring similarity. There are other measures of you can look at it as a tree or a dendrogram, there are measures of genetic similarity. I mean, there are very many such measures that in fact can be used, at least at this time.
I think we have Kyle.
Hey, thank you all for this. This is really wonderful. Just since you're in front of NHGRI Council, I thought it might be great to just hear what you all think are future research needs coming out of your work. Sort of, you identify a way forward with some suggestions, but I assume there's additional work that needs to be done. And I know you're probably reticent to advise NHGRI on specific, granting mechanisms because that gets close to policy. But just in the big picture, what are some research needs you think that the field should be addressing?
Well, I'll start by saying that there was one recommendation that I forgot to mention, Kyle, and that was our last recommendation, our recommendation number 13. That calls for the establishment of some oversight bodies or entities to help with the implementation strategies that we talked about, that we talk about in our recommendations. Recognizing that recommendations have been put forward for years with regard to the use of population descriptors, but folks have not been held accountable for implementing those recommendations. And so so we're calling on, and it may not be one oversight body, it may be different oversight bodies to monitor the progress that's being made with journals. Many journals have made policies about what researchers ought to do, and we know how those have gone. That many just have not been adhered to. And so for us, there needs to be some oversight of those processes, of what researchers are doing, bringing journal editors together to think through what are the policies and the guidances that they may do. With regard to research, we also have a recommendation about continued research that needs to happen to help us better understand the population descriptors and identify new tools and methods of assessing genetic variation and describing populations. And whether that's the Genome Institute or whomever that's going to help facilitate some of those processes. But it's going to be a range of entities that will have to collaborate in many ways to make these changes happen. Aravinda, more research needs.
I don't know that the whole committee thought of all of the ways and what to recommend to the Genome Institute in particular, but I'm not a stranger to controversy or to debate, so I'll give you my personal opinion. I think we have to think much more deeply as to who we ascertain into our studies and how that comes about. To give you a sense, I now live and work in New York, and I could go through our hospital records, clinical records, and get a so-called random sample, but that random sample is decidedly non-random. It's decidedly non-random even for New York City. In fact, it's decidedly non-random even for the island of Manhattan. So I think we have to think of, I think what we've done is sample whom we have and then ask them to describe themselves. And I just don't think that's sufficient. We really need to think of whom we want to incorporate, why, what level of diversity we want, and how we accomplish that goal. I think we need a much wider sampling of diversity, both throughout this country and in other nations of the world. How we do that it's a far greater challenge. I think there's scientific questions on how we sample throughout. Most sampling has been done through populations, but there are other ideas out there, and other ideas that have been out there from at least 40 years or longer. So there are other ways of sampling humanity. So we need to consider all of that. I'm not saying one is better than the others, but I think we need to think of how we assess human genome diversity in terms of people, not the genome per se, which we technically know how to do, and whom we incorporate into our studies, why, and what they do bring or not bring to the questions that we have in front of us. And I'm not here to give the Genome Institute and its director advice, but I don't know many other NIH institutes that focus on these questions or will focus on these questions. So it is something that this council at least should think of.
And just to follow up a little bit on that, Kyle, in terms of research needs, one of our requisites is to include environmental variables. And we're not calling on geneticists to become epidemiologists or social scientists and to measure these themselves. We know that even the social scientists are trying to figure out how to measure these, but we're calling for collaboration. So researchers, genetics researchers, researchers in genetics and genomics, who are wanting to have a better understanding of the populations they're studying and the peoples they're studying, to recognize the need to collaborate with others in other disciplines that can bring that expertise. So that, and institutions, research institutions, in many ways will need to facilitate those kinds of collaborations.
Howard?
Well, I want to say thank you to the to Aravinda and to your entire team for this really important work that you're doing. Is obviously a very complicated problem. You mentioned the recommendation is that there are different types of perhaps labels or classifications for genetics research versus other kinds of research. And the question is that do you foresee that there's going to be some sort of table of translation, so that as you go from different kinds of studies, you can actually then go back and then connect, let's say, the genetics research or genomics research to these other questions? Because otherwise, there will be studies with different kinds of labels for participants. And then, in a way, then the genetics research gets basically separated, because then we don't really know how to match up those participants to other studies anymore.
Yeah, I don't know that we have a recipe on how to do it, but basically we have to have some way of translating findings from the past in whomsoever the study subjects were and how they were described, and to do so in more contemporary terms. And I think if you look at the variety of population descriptors, things we may have called race and ethnicity sometimes can also be described in terms of, say, other aspects of culture, such as language. It could also be described in terms of geography. So I think the action of doing that will tell us that the previous labels were far more ambiguous than certain, and will also help us define what we should do going forward. But you're right, we have to have some way of translating findings and putting them in the context of what we're doing today. But I think it will take a number of examples. We didn't particularly think of specific examples or create such a table, but one of our recommendations is that we do so, because we will continue to use many of these legacy data, even though we add genomic information to them today.
Peter?
Thanks for this great work. So it seems that in some obviously race is a thing for racism, but in many contexts, race is like GC content. It's correlated with everything. And so I'm wondering, do you have a sense if we could actually capture all of the items for which race is a proxy, such as food insecurity, stress, exposures, what would be left over? What is your sense of what disease biology is actually in sort of the rest?
Go ahead.
The question about what's left over, not a whole lot. But our point in talking about not using race as a proxy is to help researchers think about, well, not as a proxy for genetic variation, but it might be a proxy for some of these social and environmental factors that we're still not able to measure very effectively. And all we're asking is that if you're going to use it as a proxy for any of those, is that you say that, right? And say what it is a proxy for. And I think your question, Peter, is that sometimes we don't know what it is a proxy for. And to be clear that we don't. I think not acknowledging that we're using it as a proxy for something is a part of the problem, because then it verily becomes a proxy for biological variation, right? I don't, I'll let Aravinda speak, because I'm not sure how to answer your question
No,
about what's.
I think you're correct. The only other thing that comes to mind is that, and by the way, race is a sort of a unique feature of American culture and history, but it's not the only such feature. There are many societies, including, in my country of birth, that caste and many other kind of orders have had similar outcomes. So the only thing that occurs to me, so it's speculative, is I would say in race there is an element of time, meaning history. And so even though we can account for many other things today, as you mentioned, I can imagine there are epigenetic and other effects that are long-term, where just income and other things might not be able to subtract away everything. And race is a particular problem. Ethnicity is a much more recent use, but is a problem simply because it's persisted so long. And these kinds of, cultural sort of norms in other societies have persisted for long periods of time. So I think that's why newer ways of looking at it is important.
Eric?
So I see the discussion is largely one, and I want to make a few comments. First of all, profound thanks to Aravinda and Charmaine for doing this, and their entire group. Now, it's not that NHGRI selected them, so I don't want to lead, unlike I selected Lynn and Gail, and so they actually had to say no to me, which they didn't. They said yes. But there's a firewall, so the National Academies actually invited them, but I was thrilled. Fence and I and others at the Institute were thrilled, not only for our co-chairs, we were thrilled and overwhelmed by the quality of the panel that was assembled. So I profound thanks to them. Second thing is, I know this was said, but I really want to emphasize it. People should really read this document. I mean, it is thick. I mean, they did not, I mean, it's a good document. This was not, you didn't cut corners. You didn't sort of just say, I'm going to give the minimum. It is well-written. It is informative. Some of you, maybe you don't need to read. I've read it, and there are a couple of things I've read a couple times. To me, it reads very educational, informative. I think it's a great thing to read and keep around and refer back to, because these are hard topics to discuss, and there's hard topics to make sure you're not speaking past each other on things. And I don't know, I found that the writing of it really helped frame the issues in a way that I personally found helpful. So I would really urge people to truly read it and not just skim it. And then the thing I would say to council in particular, and it'll relate to what I'm about to ask Vince, is, at the Institute, and I think as a community, because remember, this is not just about NHGRI by any means. First of all, this was done on behalf of 14 institutes, a large number of institutes. So I think there's lots of parts of NIH. This is very relevant. Just as genomics has spread everywhere at NIH, these issues have spread everywhere being relevant. It's not just about NIH. And it's about lots of things, about all the science that we're doing in genetics and genomics. But I think everyone involved in any aspect of this should pause and think about it. And I guess the ask I have for council is, let's not forget about this, and let's not forget about some of the recommendations. And we may get busy, we may forget, we up at the front of the table, but if in a meeting or 2 or 3 or 5, or an RFA or 2 or 3 or 5, or a new initiative 2 or 3 or 5 down the road, remind us, or bring ideas. If you think there are things we could be doing from their implementation charts, we ask council to make sure that you hold our feet to the fire, that we are properly wording things, or maybe coming up with new terminology or new approaches or design of studies. So I guess my charge is I want council to help NHGRI partner with us, please, to help think about what we could be doing in our institute to help move forward the agenda in the way that the report aims to do that. So you are absolutely welcome to the table to help us. And actually, we need that. And so I think your diverse perspective will be very, very helpful going forward. It's another reason why I want you to read it and really think about it, because I think it will require a community of funders, researchers, and advisors for us to do the right thing. So with that spirit, and to reflect the fact that we have been thinking a lot about, all right, what do we do? And when the we meaning NHGRI, but it's we meaning NHGRI extramural, we meaning NHGRI other parts of the Institute, we meaning NHGRI within the community of NIH institutes. We'll be looking to lead, I'm sure, on this, as will some of the other institutes, and all of us and Minority Health Institute come to mind. There'll be some that'll be more likely to lead, but what should we be doing? Also, what should we be doing as strong voices in the genetics and genomics community? So this is something that Vince in particular is trying to organize us for our to-do list. So I thought I'd maybe give Vince a few minutes just to go through what our list looks like, and if you think we need to add to it, we'd like to hear that. So maybe Vince, you could say a few things?
Yeah, I'll share on some things that are happening both at the NIH level and then at NHGRI. So the 14 institutes and centers that came together to fund this have been meeting since the report has come out on next steps and actions, and meeting with various parts of the NIH agency and its various components, and now inviting the other 13 institutes and centers that did not co-fund it to be part of that conversation. So there's things happening at an NIH-wide level around the conversation about the report and what is appropriate for NIH to do. Then within the Institute, we have now convened a committee. We've identified all these individuals from our extramural program, and including also individuals from our intramural program, to think about what we need to be doing as an institute and how can we lead and be at the forefront with regards to our next steps and our efforts, both from a policy perspective, but also on what research opportunities and questions that need to continue to move forward. So we will be coming back to you as a council to give us guidance, to see our recommendations and proposals, and to give us feedback. But this is something that's very active in moving forward now within the Institute. And I also just want to say thank you to both of you for your leadership and guidance that you provided the committee. I had an opportunity to watch all of the videos online and then now to see the report. So I want to thank you also for your work.
Thank you, Vince. Our pleasure.
Either of you have any last? Sorry, either of you have any last comments while you have the podium?
I will say that I trust that this report, we know it's not going to be the last, but we trust that it will be a report that moves us forward, a report that allows us to change our ways of thinking about populations, about individuals, and about how we use these variables in our research. I can't say enough about our deliberate thinking about changing the thinking, because we realize that just giving people a menu of what to do is not going to change the field and change and help us address these long-standing problems about the use of these variables. It's going to take a new way of thinking, and changing thinking is hard work. But I think if we're committed to seeing us advance, not just our field of genetics and genomics, but biomedical research and scientific research more broadly.
Well said.
Okay. And Rudy, I know we are about to go to break, which we're taking a 15-minute break. Okay. Okay, we will reconvene 10 minutes before the hour, which on the East Coast will be at 3:50 p.m. So take your break now.